in today's lecture for us to do it. So today we're doing ELISA, and the one we're doing is actually called the antibody capture method, but most scientists refer to it as the indirect ELISA. ELISA is an acronym, actually. It stands for Enzyme-Linked Immunosorbent Assay. So we're going to use an enzyme, right, denoted by that cute little Pac-Man there, linked to an antibody. Specifically, it's going to be the secondary antibody, the second antibody we use in this test today. And that's what the immunocomponent refers to. Sorbent refers to the fact that actually we're going to create a color change. And an assay, meaning we have to follow it in a sequential order, right? You have to follow the steps in the order, otherwise you won't get accurate results. So what is an antibody? It's a product of your immune system made by white blood cells known as plasma cells, which derive from B lymphocytes, commonly referred to as just B cells. They're specific to the antigens. So what are antigens? These are foreign substances that our body mounts an immunological response to, like making antibodies. So this is a specific interaction. Specific antibodies bind to specific antigens. Yes, we have self-antigen, but we don't usually respond to ourselves, right? It's when it's foreign. So the other thing that we're going to utilize in this test is what's referred to as an anti-antibody antibody. Wow, that's a mouthful, right? So a lot of times, instead of saying anti 3 times, right, we use one of the synonyms for antibody. Immunoglobulin actually means antibody. It's another name for an antibody. So we say immunoglobulin antibody. And again, this means it's an antibody against an antibody. So we're dealing with an antibody today that binds to other antibodies. Sometimes when you're dealing with human, you'd even say human anti-antibody. So this is the general structure of an antibody, and those of you guys in my lecture class should know the answer to this, right? Where do the antigens bind? Yeah, A and B. The variable region of the light and heavy chains. Where could an antibody bind then? If this is sticking to the antigens, this portion of the antibody, what part is the anti-antibody going to bind to? Yes, the FC region down here. So the antibodies are different colored here. Why? Is every antibody the same? No, each antibody is unique, right? So in a person's serum, if we were to take their sample, we'd have lots of different antibodies denoted by different colors here. And that's the region that's different, right? That variable region that binds to the specific little piece on an antigen. And you actually produce 5 different classes of antibodies, something we're not going to go into in this class, but we talk about in lecture. So when I used to do these types of tests in research, you can run them on what's referred to as plasma, when the blood has been taken with an anticoagulant. And when you separate out the red blood cells from the liquid phase of the blood, this is called plasma. Immunologists like myself do not like plasma because plasma contains not only the antibodies, but it also contains the clotting factors that were not used because the blood was not allowed to clot. These excess proteins can get in the way of these types of tests, right, and cause problems. I prefer, and most immunologists would prefer, to work with serum samples. But that means the blood, you have to take 2 blood tubes from your patient if you're running other tests that you need non-coagulated blood flow. So in this case, you would take this tube, and this tube allows and actually promotes the blood to clot. So all the clotting proteins end up in the clot. And that liquid phase you have is called serum. And serum, right, contains the antibodies as well, but not the clotting proteins, right? So we do a lot of serological tests, and that's what they're referring to, a serum sample. And typically, again, we're looking for antibodies in that serum, in that liquid phase of your blood. So what are we doing today? Are we looking for an antibody in someone's sample or an antigen? Antibody. We're looking for an antibody. We're doing an indirect, not a direct ELISA. Right? So Bio-Rad has animations, right, which I added the link to for you guys right here. There's no audio to this, right? What? Oh, here we go. I don't know why I wasn't loading something about, I think it's bad. It's not bad. Right, so we want to click on antibody detection, right? So this is what you guys are going to do, right? You're going to start out with plastic wells. So go ahead and look in your bin. You have a strip of plastic wells. Somebody get up and get some Sharpie markers, one for each table, please. The first step you're going to do is you're going to actually add antigen, right? I'm just going to quickly go through this, and then we're going to go step by step. So this is the overview. The antigens are going to stick to the wells. You're going to wash out anything that doesn't bind, and I'm going to teach you how to do the wash step. Then you're going to add the primary antibodies. The ones that bind to the antigen will bind. The ones that don't, we will wash away. Then we're going to add our secondary antibody. This one has the little enzyme Pac-Man there attached to it. It will bind to the first antibody, the primary, right? This is the anti-antibody. We'll wash away anybody who doesn't bind. Then we're going to add a substrate that I have in the refrigerator. That substrate's going to interact with the enzyme. It's going to be colorless, and when it interacts with the enzyme, if it's present, it's going to turn blue. Right? And that tells us everything is in that tube, right? Antigen, antibody, secondary antibody with the enzyme, reaction happens. That's a positive result. So what's the difference between that? Let's see, how do I, oh, here we go. So I can go to the antigen detection. What do you think is going to be different here? So our sample is going to be containing different stuff, right? The antigens and stuff are going to bind to the well if you were to do antigen detection. And then we use a purified antibody that only binds to the antigen, right? So we would add that in. That's purified. Notice they're all the same. They only bind to the antigen. Anything that doesn't bind gets washed away. But can we see this? No, we need that secondary antibody, that anti-antibody with the enzyme linked to it, right, for this test to work. So then we add it in, right? We add in our substrate after we wash. And then we get a color change if everything's there. So we do the same thing. It's just one of those steps. Are you using a patient sample, right? Are you using purified antigen or antibody? So let's go back to our test. Helps. I don't know why my button's not there. It's driving me crazy. All right. So what are types of antigen tests? So these are if the person has the antigen. So here's some examples. Rapid strep test is looking for the antigen for the bacteria that causes strep. When you ovulate, what do you produce? Luteinizing hormone. Could that be considered an antigen? Can we use an antibody that recognizes it? You betcha. That's how these tests work. What do you produce when you're pregnant? Human chorionic growth hormone. That's the antigen. Guess what we use to detect it? Specific antibodies. Drug tests. HIV antigens. Flu tests. Looking for the antigen. These are all direct ELISAs, y'all. We're using antibodies to detect the presence of the antigens. Indirect ELISA, we're looking for what? Antibodies. So what would you make antibodies against? Why would we want to know if you made antibodies? To see if you're vaccinated, right? When they were running titers, right, they're actually quantifying how many antibodies you have against, say, rubella, German measles. But we also use it in a variety of ways. HIV antibodies, heartworm antibodies that your dog produces when they have heartworm, hepatitis viruses, and then again, any of the things that we vaccinated people against, right? They use this type of technology to see, do you have that specific antibody we're looking for? So with HIV, right, it affects the CD4 T cells. So as that progresses, we could detect the levels of the CD4 T cells, and when they get really low, you're considered to be immunodeficient. And in that progress, we can also detect the presence of the actual virus, right? The RNA from the virus. And you can see it spikes in the beginning, and in the latency period kind of levels out. But when T cells are completely destroyed, it'll spike again. So this is one of those tests that they test, blood for very quickly and easily to determine if it's got the antigens or the antibodies to this. So we're going to create an antibody sandwich, right? We're going to start with an immobilized surface. Those are your little wells that you guys have. We're going to add the antigen, purified antigen, in this case, a secondary antibody, right, which your patient may or may not have, because that's what we want to know, right? Do they have this antibody that binds to the antigen? Then we're going to add a purified secondary antibody, which has what linked to it? The enzyme, right? The enzyme will convert the substrate into a color product when we add it. Other tests, they'll use actually fluorescent things and chemoluminescent products. So here's some examples of where they actually used it to stain cells on a slide. That was the immobilized surface. And then they add the antibodies with these fluorescent markers on them. And so they're looking for specific proteins and where they're located in the cell. So that the nucleus is stained blue, but look at where these proteins are. And these are all spread throughout. But these are just on the edge of those cells, right? So I was really able to tell them, what's going on by being able to label, right, these specific proteins for these cells. So this is used a lot in research, not only for detection, but also in this case for labeling. So let's take our microtiter strips. So you have a little strip of wells, right? And we're going to label them. With your Sharpie marker, you're going to write right on that strip, and you'll see that there's a little white demo in there, right? So you're going to go, I think, plus 3 times, negative, 3, 3, 4, 4, right? So inside your bin, there's the example right there. Yeah. Yeah. So you want to label your strip just like the demo. And you write right on the tube, right on the wells as it is in the demo. And then you have another rack, right, that you can set that rack in that doesn't have a rack or strip. So you have another rack in there, Jess. You should have another white rack for holding your strips. Yeah, there you go. So flip that over and then set your strips. Yeah, there you go. It'll keep it from getting knocked over. Okay. The next step is going to be to add from the green tube that I'm going to give you guys in a minute. It's in those racks back there. Purified antigen. And we're going to add 50 microliters. So remember, you've used these fixed volume microliter pipettors before, and we got to put the little yellow tips on it. So you'll notice that the little yellow tips, just so that we don't keep getting up and go to the trash can, you can put in the jar where your pipettors are. Yeah, so I'm going to go ahead and pause. So we're not going to do it right away because technically you're supposed to wait about 5 minutes, right? But we'll wait as long as it takes me to talk through this next section. So the next thing I'll show you guys after we talk about this next section is you're going to do wash steps. So the first thing is we're going to actually dump out the liquid, and then we're going to add wash buffer. And this is going to remove anything that didn't bind, right? Any of the antigen that didn't bind before we do the next step, which is adding the primary antibodies, which is going to be when we're going to follow the code you guys wrote on those tubes. So we're going to wash twice, right? And we'll do that after we talk about this. So why do we do washes? So what happened in that animation when we washed? What got washed out? Anything that didn't bind, right? Anything that didn't become immobilized. Because we want it to stick. So anything unbound, right, gets removed. So what would be the consequences if we wash too vigorously, though? We'll remove bound antigen, and we don't want that, right? Because that would end up with false negative results, right? Because we don't have anything sticking. What if we don't wash enough, though? You're not really going to get false positives, right? But you're going to get competition here, where stuff that isn't bound is going to soak up the stuff we put in and prevent it from sticking. Right? It'll soak it up. And this will affect the sensitivity of our test. We will probably still get positive results, but they'll not be as strongly positive as we would have preferred. And this test can be done quantitatively, which means we could actually put it on a spectrophotometer and get a number for the amount of color that is generated. We're not going to do that today. We're just going to see if we get positive or negative, right? We're just going to be qualitative instead of quantitative. We're not going to get a number. Okay. We're not going to do the blocking step. We don't need to. Our wash step is going to act as our blocker, which means it's going to take up all the extra binding sites so that stuff doesn't stick where it's not supposed to. So then the next step after we wash is in the plus wells, you're going to add the antibody, right? These are our positive controls. Why would you want to do a positive control? I just answered this one though, right? What are we putting in there? What are we testing for today? The primary antibody, right? That's what our positive control is going to have in it. Why are we doing positive controls? Yeah, make sure our test results are accurate. We know what they look like. That we perform the test properly, or the test itself is working properly, right? Does the pregnancy test have a built-in control? Yeah, it's why you get 2 lines when you're pregnant. One is the control well, the control line. The other is your test sample. 1 line, control worked, you're not pregnant. 2 lines, control worked, and you're pregnant, right? I was so excited I took a picture of mine.
The positive control is the primary antibody?
Yes, primary antibody. So we want to make sure our test is working properly, right? What are the possible causes of negative results in our positive controls? That's not good, right? In the case of the pregnancy test, it's going to tell you to throw it out and do a new one, right? Our test results are null and void, right? If we get negative results for positive controls, yeah, we maybe washed too much, right? We maybe added the wrong reagents. Maybe the reagents are expired, which is usually what happens with the pregnancy test. There is an expiration date for those things. Make sure you look at it. So our positive controls, as you can see here, right, from that animation, there's the antigen, the antibody's going to bind, right? The primary antibody. Negative controls. So does this contain the antibody? If it's a negative control, we want a negative result. Does it contain the antibody? No. Right? It's lacking the primary antibody that we're testing for today. And this is to help us prove, right, that without that antibody, this test should be negative. Right? So it's just buffer solution that I'm giving you guys. No antibody in it. So what happens if we get positive results in our negative wells? Is this good? No, some type of contamination, something went wrong, right? Failure to do a wash step, right? If you don't wash them out, that might be the problem. You might have cross-contaminated, and this is why it's important when I show you guys how to wash that you do it the right way so you don't contaminate your wells, one to the other. How would positive result in negative controls affect our interpretation of our results? Could we use those results? No, right? It invalidates our experiment. Right? Is that person really positive or negative? We don't know. Our controls didn't come out right. Got to start over. Because you don't want to tell somebody they have HIV if they don't, right? You want to be sure. So our negative controls, remember, should just have the antigen. No antibody is going to attach, right? No antibody should be present. It should look like this. Right? Just antigen, no antibody. So we're obviously going to run a test today. You have two patients, Mr. Number 3 and Mr. Number 4. You can pretend you're testing for whatever you want to test for, whatever antibody you think is cool. One of these guys is going to get good or bad news depending on what you're testing for, right? And the other one is going to be the opposite result. Your challenge today is to find out who is positive and who is negative for the primary antibody. So we're going to do this in triplicate. So notice you wrote these numbers and these symbols 3 times. Why are we going to do that? It's more accurate and more precise, right? If we do something 3 times, what's the chance you're going to make a mistake? Less likely, right? Than just doing it once. So again, all 3 should come out the same if we did the same thing 3 times correctly, right? We want to be here, highly accurate and highly precise. We want them to all be the same. What if we get inconsistent results? Is that good? Right? So what if one tube of the triplicate is positive and the other 2 are negative? What does that mean? You probably made a mistake, right? Are you going to trust those results? No. It could be poor technique. It also can be poor sensitivity too. But this is a pretty sensitive test that we're doing today. So more likely you screwed up. Right? Variability in our replicates casts doubt on our outcomes, right? This is not good. So consider our patients, right? As I said, somebody has the antibody and is positive. One of them does not, right? No antibodies, no binding. There'll just be antigen in that tube. Okay? So it can be quantitative because notice the different variations of the color. And as I said, we could put this plate on a plate reader and we could read and actually get a number for it, but we're not going to do that today. I hope to do that in the future. And that's what I actually did when I ran these tests on the armadillos to find out how much antibody they had against leprosy. I would actually quantify it. I would run a positive control that was a dilution. So as you can see, it gets darker as there's more antibody present. And so when I did the test samples, I'd get different ranges of color. And with the spectrophotometer, I'd actually get a number for the amount of color in each one of those wells. And I could calculate about how many antibodies, a titer for them about how many antibodies they had in the sample. All right, so let's take a moment and add our individual samples.